Dear students, now I am going to discuss about the topic history of immunology. This topic belongs to your course Immunology Module 1. So the term immunity was derived from the Latin word uh, immunis, that means exempt, and its actual English meaning is protection from disease, which was originally referred to as protection from legal prosecution offered to Roman senators during their tenures in office. This term was adopted subsequently to designate the naturally acquired protection against diseases such as measles or smallpox. And it was indicated that an individual can develop uh, lifelong resistance to a particular disease after having contracted it only once in his or her life. So the cells and molecules responsible for immunity consists the immune system and their collective and coordinate response of the body against the foreign particles. It may be some microbes, it may be some toxins. Uh, is called the immune response. This immune response can be of two types. It may be specific, it may be non-specific. So the concept of immunity has existed since the ancient times, and we can find an example in Chinese customs of making children resistant to smallpox by making them inhale powders made from the skin lessons of patients recovering from the disease. This is an old concept. The first European mention of immunity uh, was recorded by Thucydides in Athens during the 5th century BC. And he wrote that only those who had recovered from plague uh, could nurse the sick because they would not contact the disease for the second time. Once the concept of existence of immunity was established, it was not long before manipulation of immunity under control conditions. First, it was uh, the scientist known as Edward Jenner. Jenner, who in a successful experiment injected the material from cowpox pustule into the arm of an 8-year-old boy and demonstrated the lack of development of disease after subsequent exposure of smallpox. And for his novel work, he has been regarded as the founder of modern immunology. This was based on his observations that milkmaids who had suffered from cowpox never contracted the more serious smallpox. His technique of inoculating with cowpox to protect against smallpox spread quite very quickly throughout Europe. However, for many reasons, including lack of knowledge of obvious disease targets and their causes, it was after nearly 100 years that this technique was applied to prevent smallpox. The experimental work of Bering and Kitasato in 1890 gave the first insights into the mechanism of sorry, into the mechanism of immunity. And for further insights, von Behring got Nobel Prize in 1901. So von Behring and Kitasato demonstrated that serum is a liquid non-cellular component of coagulated blood from animals previously immunized to diphtheria could transfer the immune state to unimmunized animals. Since then, immunology as a field of study has come a long way, and it has been and remains one of the hottest field in research, as shown by the statistics of about 17 Nobel Prize have been awarded to scientists involved in immunological research. So next one. So in this process, Louis Pasteur, ex pioneer, has done pioneer work against vestibular testis as plague, to refer to the injection of 18 culture of vaccine in honor of Jenner. And he applied the same technique for anthrax also, using bacillus anthracis and also with rabies. These are his novel work. Then in 80s, Metchnikoff discovered phagocytic cells that actually ingest the microbes and the particles and conferred as cell-mediated immunity. In 90s, uh, the noble work of Behring and Kitasato came that they has discovered that serum. So it's not a cell-mediated one; it's also a liquid or blood-mediated immune system. So both cells and serum or blood contribute to immunity. And in 1930s, early techniques made it easier to study humoral element. Uh, and actually, the scientists have discovered that the active component of blood, not the blood cell, it's a protein molecule that is known as gamma globulin protein. Gamma globulin protein. So in 1950s, the discovery of two types of cells, B cell and T cell, links lymphocytes to both cellular as well as humoral immunity. And last few years, scientists has evolved uh, so much of uh, activity, has evolved so much and engaged themselves in so much of activity in development of different type of antibodies, mostly focusing on monoclonal antibody for the therapeutic purpose. And they have revealed unlimited range of reactivity and also conjugated with newly synthesized chemicals. And along with these newly synthesized chemicals, the antibodies can be automatic choice for the practitioners for the treatment of deadly diseases like cancer. So one of the pioneer of immunology is Paul Ehrlich. He has developed a series of tissue staining dyes, including that for tubercle bacilli. He has worked with Robert Koch and developed antitoxin, diphtheria toxin, and his side chain theory of antibody production and antibody formation, uh, antigen stimulated receptors, and he has said that surface receptor bound via lock and key model. And for his pioneering works, he has got Nobel Prize in 1908 along with Mexico. One more renowned scientist in this field is Susumu Tenigawa, and his pioneer work is for the cloning of immunoglobulin gene. In 1987, he got Nobel Prize for this discovery of genetic principles of generation of antibody diversity. So this is not the list, but Peter Doherty and Jim Kernagel. These are the two scientists. They have got also Nobel Prize in 1996 for their discovery of concerning the specificity of cell-mediated immune response. So immunology has evolved as a science, as a subject of science, and it is the study of molecules, cells, organs, and systems responsible for the recognition and disposal of non-self material or the foreign material from the host. So it's a study of mechanism that protects us, the individuals, from either injury, from infections created by exogenous microorganisms like bacteria, fungi, and viruses, exogenous chemicals, say pollens and toxins, poisons, endogenous cells like malignant cells. So, immune, what is immunity? Immunity is a term. It is used for the resistance to a particular disease causing uh, caused by the infection of an organism or any other harmful substances that is called as toxin. So there are two types of immunity. One is the uh, active immunity, another one is the passive immunity. So active immunity is induced by the exposure to a foreign antigen called active immunity. Active immunity is a resistance developed by an individual after contact with a foreign antigen, maybe a microorganism. That contact to be in the form of clinical or subclinical infection, uh, immunization with liver, with liver or killed and infectious agents or their antigens, or else exposure to microbial products such as toxins or toxins. In all these circumstances, the immune system of the host is stimulated to elicit an immune response consisting of antibodies and activated helper T cells, along with cytotoxic T lymphocytes. So, active immunity develops after a latent period during which immunity of the host is geared up to act against the microorganism. Hence, it is slow in onset, especially during the primary response. However, once its active form of immunity develops, it is long lasting, and thus major advantage, that is the major advantage of active immunity. Active immunity can be of two types: natural active immunity and artificial active immunity. So natural active immunity is caused by or acquired by natural clinical subclinical infections. Artificial one is induced in individual by vaccines. So this is artificial active immunity. These are the different types of immunity. So mostly uh, classified as non-specific one is the natural one or the innate immunity, and the acquired immunity or specific immunity or adaptive immunity. So innate immunity is a resistance that an individual gets uh, through birth. Innate immunity can be classified as individual immunity, racial immunity, and species immunity. So, for example, if one homozygous twin develops tuberculosis, there is a very high possibility that the other twin will also develop tuberculosis. But the heterozygous twins, there is a very low possibility of the other twin suffering from tuberculosis. So, this innate immunity, yeah, the interferons, spleen secretions, phagocytosis, reticular endothelial cells, inflammations, natural killer cells, complement system, they are actually taking care of this innate immune system or the natural immunity. Next one is acquired immunity, that is a specific immunity. So acquired immunity or adaptive immunity, since the potency of immune response is acquired by experience on. Acquired immunity can be of different type. So it may be active, it may be passive. So this particular immunity against a microbe may be induced by the host's response to the microbe or by transfer of antibodies or lymphocyte specific